Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. We invite you into the stories, struggles, and breakthroughs shaping the future of human health. From cutting-edge brain science to discoveries transforming how we heal, adapt, and thrive, we explore the ideas that matter and the people behind them. Together, we look across diseases, technology, and research fields to understand not just what is changing, but why it matters for patients, families, and the future we all share. Join us as we uncover new insights, spark collaboration, and illuminate the science that can improve our lives. Welcome to Research Renaissance. I'm Deborah Westfall, your host and guide through the fascinating future of health science. Joining me today is Dr. Rachel Bennett, an associate professor at Massachusetts General Hospital and Harvard Medical School. Rachel is also a 2024 and 2025 Toffler Scholar. Her research is helping reshape how scientists understand the relationship between brain blood vessels, aging, and neurodegeneration. So welcome, Rachel.
Hi, thank you. It's good to be here.
So let's just kind of start with stepping back and historically looking at some of the Alzheimer's research that has been, kind of in the past. And maybe I've got this wrong, but they've treated blood vessels as secondary damage to what was going on inside the brain. But your work suggests that maybe this is not necessarily true, that some of that vascular dysfunction may be intertwined with some of the tau pathologies itself. Can you talk about that? And are we witnessing something that is very different in how we think about neurodegeneration?
Yeah, that's exactly right. And that's a great place to start because you're right. Historically, if we go back even to some of the earliest writings about Alzheimer's disease, 100 years or so ago, they were describing vascular changes in Alzheimer's disease. But it was kind of set to the side because the thing that jumps out to you when you look at pathology in an Alzheimer's disease brain is amyloid beta plaques and tau-containing neurofibrillary tangles. And, there's been a lot of work over the years looking at vascular function and how it changes in disease. So there have been some really excellent longitudinal studies where people have looked at cohorts of, 5,000 individuals and tracked them over up to 7 years or more. And they can see that cerebral perfusion is reduced just at baseline in people who are at a higher risk of developing Alzheimer's disease later in life. And I think it's been estimated even that 50 to 80% of all Alzheimer's disease cases that come to autopsy end up having vascular abnormalities in some shape or form or another, in addition to plaques and tangles. And so there really is just this tight link between vascular function and just risk of developing Alzheimer's disease. But I think largely the field has kind of ascribed that to, it's just worse to have two diseases than one. So if you're at risk for cardiovascular disease, that's going to put you at risk for a whole host of other disorders. And one of those is Alzheimer's. And so largely, the focus hasn't been so much on, reversing vascular pathology. It's been in reversing amyloids. Our best therapeutics currently are targeting amyloid beta plaques, and they're really good at clearing plaques in the brain. But they don't necessarily reverse some of these vascular changes. And so what our work did, and we published this, I think, a couple of years ago now in Acta Neuropathologica, is we were looking at tissues in a new way. So instead of looking at traditional thin tissue sections, we were looking at very thick sections. So when I say thick, I mean not particularly thick. So these are about 1 millimeter. So if you take a centimeter, we're looking at 1/10 of a centimeter, essentially thick piece of tissue. And for that was sufficient to actually see vascular networks and all the vascular structure when we do some immunofluorescent stains. And what jumped out to us when we did that was that tau is accumulating around blood vessels in a way that no one had appreciated before, because when you look at a very thin section, you don't see that three-dimensional structure, and you miss a lot of the pathology and a lot of the features of pathology that we're able to pick up in these thick tissues. And so that was really novel and really makes us kind of rethink what's going on. And, another thing that we came across is an experimental model of disease. We were looking for tau, and we were using a model that develops tau neurofibrillary tangles. And what jumped out to us was that blood vessels were remarkably altered in their structure. So the blood vessels that we saw were spiraled. They had corkscrews. There was too many of them. If you measured the overall length of blood vessels in this system, there was just too much vasculature for the amount of brain tissue that was present. And that was really odd because all that this experimental model had was tau pathology. So, we're kind of you can't do that experiment in people, obviously, because, there's always the other factors. So maybe this individual had diabetes or was a smoker and did have that increased cardiovascular disease risk that was causing vasculature to change. And that was just comorbid to developing Alzheimer's disease. But this experimental system, this did not have, these were not smokers. They did not have diabetes. The only thing that they had was tau. And so that kind of, to us, indicated that tau pathology alone was sufficient to drive vascular dysfunction and changes in vascular morphology. And that's really novel. And that's not a way that we've been looking at disease.
Yeah. This is really interesting. What made you guys step back and look at this thick tissue? Because, it was a practice. It was a process that this is how we prepare the slides and this is how we do it. What was that kind of moment that said, hey, what if we took some more? I mean, because that's innovative, and that's out of practice. Was there a reason why? Was there a hypothesis or for this?
Well, frustration, perhaps.
Okay.
So when you start asking questions about blood vessels, it rapidly becomes apparent that looking at a thin tissue section is not sufficient. You end up with tiny little cross sections of blood vessels, and you don't have a lot of information. So we really like to think about those vascular networks and how, the artery is connected to the capillary network, and, there's primary and secondary and tertiary structures, and then that all builds up to the venule level, and then that drains out of the brain. And, you can't see that if you just have a single cross section and a single plane to image. So it was actually an older technique. So in the past, people had been doing this. They were using different dyes and stains. I think it was India ink that you could inject. You had to do a very special preparation and prepare the tissues just for this. And then you'll find some older publications from several decades ago where they did this. And they actually found very odd blood vessels and Alzheimer's disease tissues as well. But, it was such a difficult thing to do that no one really followed up on it. So it was like, okay, blood vessels are kind of odd in Alzheimer's disease, and maybe it has something to do with Alzheimer's, and maybe it doesn't have anything to do with the disease. But just a little side note in research history. And so, we kind of went back, and there's some newer methodologies that have developed over the years where you could do this in a fluorescent preparation, so not using these laborious dyes and stains. And it's kind of funny because I think when I started way back when I was a postdoctoral researcher, this was one of the first things that I tackled. I thought, here I'm gonna visualize all the blood vessels in the brain, and it's gonna be so cool, and we'll be able to relate all these features to disease, and it's gonna be great. And we could do it technically, but once we had these images, they're terabytes in size, and so going back 10 plus years ago, we literally did not have access to computers that had enough storage space or RAM to even open the images. And so we're kind of ahead of the time. And so it took several years for that to really come to fruition. We're able to do these experiments in the way that we wanted to. And now we're able to do that, across many tens, if not hundreds of samples.
So do we have the capability now to look at all the vascular networks inside the brain? I mean, do we have that ability with new computers and imaging and all of this to be able to really map that out and understand it?
Yeah, if you'd asked me even a year ago, I would say, I don't know, but I think we do. I think we're getting to the point where really we can start to study every single blood vessel in the brain and resolve it at the cell level and capture changes that we wouldn't have been able to, just a few months ago. I think the last imaging session we did. So this is all done. You take these tissues, you put them through a chemical process that removes some of the lipids so you can actually visualize throughout the thick piece of tissue, because lipids are very light scattering. So if you don't remove them, you actually can't see through the tissue. And that kind of makes sense because, I think we've all seen, even just your skin, you can't see through it very well. So you have to treat it in a way where you can actually get light to pass through it. And so what we have done is cleared these tissues of lipids, labeled them with some fluorescently tagged antibodies against the proteins that we're interested in. So proteins that form the structure of blood vessels, or maybe to tau to resolve tau pathology or amyloid beta, you name it. And then we put on a light sheet microscope, which is, I think, just the coolest thing. It's a microscope that actually forms a sheet of light. There's 2 different objective lenses, and it allows you to scan through very rapidly a very thick, large volume of tissue. And from there, we can kind of 3D reconstruct everything that's going on. And so in this last imaging session that we completed, I think we were able to trace 47 meters of blood vessels. Wow. And that was in a single gyrus. And we were looking at a piece of human brain tissue. And a gyrus is, all the folds of the brain. So we're looking at just a single piece of a single fold in the brain that had 47 meters of vasculature. So we're really able to scale this up now to the point where we can start to think about, the 500 plus miles of vasculature that are in the human brain.
That's amazing. That's amazing. And, that network, along with all the neural networks and everything in there, it's like, who would have known? And that's really incredible. I'm also kind of wondering, you talked about this crazy vascular kind of distortion with the curls and the spirals if it was around tau. Does vascular in other places in the body, does this happen for other reasons? Is this pretty unique in the brain?
Well, that's a great question. Yes, it does happen in other places. So notably in tumors, you get very odd-looking blood vessels that are produced from angiogenesis processes. So you get usually too much vasculature, and you end up with a lot of these kind of corkscrew type appearing vessels that are highly branched and not particularly functional. So they're very bad at doing their job of delivering blood flow to the tissue that they're located in. And also in hypertension, you tend to see things like corkscrews appear. So I suspect there's an interesting intersection here between mechanisms where we're seeing tau can kind of independently, or at least in the setting of tau pathology, it can independently exert this effect where we're getting changes in vascular morphology. And then some of these kind of systemic factors that we know contribute to disease progression can also induce those same vascular changes. So I think the more we learn about this, the more it's going to make sense why some of these vascular risk factors really do contribute to disease development.
I think I heard somewhere that we have found tau in other parts of the body. Is that true? Is it flowing through the blood and getting to other places in other organs or other places in the body?
Yeah, tau is a mysterious protein. So it seems like the harder you look for it, the more you see that it's everywhere. So in the brain, there are 6 isoforms of tau, so different flavors of tau that are produced by the gene. And they're predominantly expressed in the brain, and then peripherally, there's this big tau, which has a bunch of extra amino acids attached to it. So it's much larger, so we call it big tau. And it's made by peripheral neurons mostly. But there's also things like cardiomyocytes that produce like a little bit of tau expression as well, and no one really knows exactly why. And then it's important to note also that a lot of the tau that's produced in the brain can also be detected in plasma. So that's the basis of a lot of these plasma biomarkers for Alzheimer's disease that are being used clinically now to detect changes associated with disease development.
Yeah. If you could just kind of explain then from your perspective, where the science is in understanding the vascular inside the brain, and then through the blood-brain barrier to the larger body. What are we finding out? Like you just said that is that we're finding that the brain is connected to other parts and other organs and through other systems. What is your perspective of kind of what that understanding is today?
Yeah, well, I think, I was trained as a neuroscientist, so I did my PhD work at Washington University in St. Louis in their neuroscience program, and it was phenomenal. And, a lot of what you learn when you're in those programs is very neuron centric. And so, blood vessels and endothelial cells, which make up the structure of blood vessels, might be a little footnote in your neuroscience textbook. And so most of what you learn is not about kind of these other cell types that are very important to supporting brain health, but not typically a focus of neuroscience generally. So I think we're learning more about how important these other cell types are in not just supporting neuron health, but also in disease development. So, I think there's a role for these and specific gene expression changes that are taking place within things like endothelial cells that could be contributing to even things like tau pathology. And tau clearance in the brain is also a very active area of research. And so a lot of those clearance mechanisms are along blood vessels. So there's perivascular clearance routes that help kind of remove wastes from the brain. And the more we kind of focus and learn about those, the more we can kind of understand what's the natural history of tau protein in the brain and how is it cleared and how does disruption to that clearance contribute to the accumulation of tau in neurons.
Yeah. So what got you into neuroscience? What was that? You had lots of choices, I'm sure, to pursue what you were going to pursue as a career. What was that sent you to University of Washington to study in St. Louis to study neuroscience?
Yeah, there's one memory in particular that really sticks out. And it was just happened to be a high school field trip that we took where we went to a local brain bank. So it's a repository where people who donate their brain to science, the tissue comes there at autopsy and then that's distributed to researchers really across the country. And for some reason, I think our science teacher decided, I think it'd be fun to take a group of high school students to the brain bank and just go see what's going on there. And so that's what we did. And one of the researchers, kind of at the end of his presentation, had everyone come over to a microscope and look at a slide. And the slide was it was a Alzheimer's disease piece of cortex that had been stained to reveal amyloid beta plaques. And I think for me, that was just like, so, here you can actually see and visualize disease. And before it was just Alzheimer's disease. It's something that happens to your brain. And there I could visually see that it was, a pathological change that was occurring. And it made it more approachable and like a problem that we could solve if we just could understand and really visualize what was going on in the brain. And yeah. I think from that moment, it just felt like a interesting kind of area for me to go because I love understanding how things work. And I love to try to, pick things apart and solve puzzles. So yeah. It gripped me.
Good for that teacher. That's amazing. That's what teachers should be doing is opening up those doors and have you ever gone back and did you ever go back afterwards and tell your teacher that was the moment?
No, I really should though.
It's like, thank you, right? That's amazing. And I think I hope that you get this correct. You originally trained in a traumatic brain injury clinic or in some area to look at traumatic brain injury and really that immune system and what does that mean to, the brain and the functioning. How did I get that right? And then how did that lead to the Alzheimer's kind of focus?
Yeah, so my PhD work was done in the lab of David Brody. So he's a neurologist. At the time, he was at Washington University in St. Louis, and now he's since moved on. He runs a specialized traumatic brain injury clinic at the Uniformed Services University. And yeah, the work that we were doing was looking at a model of repetitive concussion. And I was really interested in it because there's these long connections between neurons. They're called axons. And I was just fascinated with how they work because, some of the longest axons are a full meter. They stretch from your brain all the way down your spinal cord. And the fact that, there's these connections that can kind of facilitate communication between cells at such a great distance and so quickly, just for some reason struck me as an area of high interest. And in repetitive concussion, these are the connections that tend to get disrupted by head impact. And so they physically, mechanically stretch the axon and cause it to break. And that process is a little bit mysterious in that it occurs and it can either resolve or the axon can be just kind of irreversibly damaged, and it leads to eventual degeneration of the neuronal soma. And so we had a model where we were trying to understand this process a little bit better. And it's a largely invisible lesion. So these axons are extremely small. They're maybe about 200 nanometers wide. I think the range is 50 nanometers up to a micron wide, and in a lot of animals and in people, they can be a little bit larger. And so that means that, again, how do you see them? So an injured axon that's 200 nanometers wide is technically below the limits of light microscopy resolution. So again, if you go in with whatever you want to stain it for, you're really not going to see much of anything. And so someone in the lab before me had come up with this process. It was actually an old method called silver staining that just kind of binds to aggregated proteins and makes them visible. And when he did the silver stain, you could see that in these concussion models, there was a lot of axonal injury, and it persisted for a really, really long time, like well beyond when you think injury would be resolved. And I was fascinated by why that could be. And so my work was looking at how the inflammatory cells in the brain, microglia, contribute to that kind of chronic degeneration that's seen in repetitive concussion. That's really important because, people who end up with head injury are at not only greater risk for Alzheimer's disease, but the more they accumulate head injuries, the worse outcomes they tend to have. And so trying to understand the pathophysiology of those processes and how we can kind of speed up recovery after injury is really important.
Yeah. Is there any connection with that and what you're looking at then with the vascular system? Is there any connection there, do you think?
Well, again, it's kind of funny, the themes that keep coming up in my career, because yes, there absolutely is. At the time when I kind of made the transition from traumatic brain injury to an Alzheimer's disease lab as a postdoctoral researcher, my connection was tau, but blood vessels keep cropping up in my career. So in traumatic brain injury, the worst end of the spectrum, these individuals who suffer multiple concussive hits end up with chronic traumatic encephalopathy, which is a disease characterized by the accumulation of tau at the depths of the sulci, so where the folds meet in the brain. Mm-hmm. And primarily the tau accumulates around blood vessels, which is thought to be primarily because those are sites of the greatest mechanical shear stresses. But now that I kind of thought more deeply about blood vessels and Alzheimer's disease, I'd love to go back and rethink that, because I think blood vessels might be unique sites of developing tau pathology kind of early on.
Yeah. Isn't that something? I mean, and it's really interesting when you said, at the time that you started, it's got to be amazing how fast the science is moving in a lot of ways, but in some ways, maybe not as fast as you would like it to move. What are you, just from your career and the time that you've spent, can you give us some idea of how fast? Because a lot of people would say, well, why don't we know? Like, why don't we know? We've spent so much money. We've been looking at this for so long. Why don't we know what causes this? Or why don't we know why it happens more. But it is moving fast. What's your perspective on all the changes that are happening, and what do you feel for the next 5 or 10 years?
Yeah, I mean, I think the real question is, what can't we see? And that's really the limitation. I mean, I told you about the invisible injuries of concussive head impact. And, similarly, when we think about tau pathology in the brain, so it's thought that tau correlates really well with neuron loss. So if you count how many tangles are in the brain, it matches up really well with how many neurons are lost. But we don't really know what it is about tau that leads to the loss of neurons. And, I think that's kind of one of the main challenges is how do you kind of visualize what disappears? So I hate to be a little obtuse about this, but, the biggest challenge is that we don't know why neurons die because it's impossible to study a dead neuron. It's just fundamentally gone. So we really need better ways to visualize the processes that lead up to that and kind of the intermediate steps. So if there's something that comes before a neurofibrillary tangle, how do we see that? And what method do we need to see that? And so a lot of the challenges that we're facing, I think, as a field is just coming up with new ways to look at disease and kind of new ideas that are beyond just amyloid beta and tau alone.
Yeah. Yeah. Your work today focuses on those really, really small blood vessels. And then I would think that there's a lot of work in that imagery that needs to happen to really understand what is it that we're not seeing. Are we there yet with some of the technology, or is there a big gap there that needs to be filled?
Yeah, I think we've gotten to the point where we can visualize, individual blood vessels, individual cells within blood vessels quite well. But you don't know what you don't know. So we tend to think about, just in the way that we think Alzheimer's disease is a disease of tau and amyloid beta, we think of imaging things like proteins before we think of imaging things like lipids and carbohydrates. So obviously there's more than just protein in your brain, and most of your brain is lipids. So, we're not even really thinking about lipids as a field. I mean, there's certainly some folks that are starting to go that way, but there's just a lot of unknowns, and it's challenging to think of what you even do with all that data. So I mentioned how we have these 1 terabyte images of, relatively large, but still quite small compared to the entire human brain, pieces of tissue. And it's hard to know, like, once you have collected all of this data, how do you store it? How do you analyze it in the best way? And so really just building out a lot of those analysis tools, I would say more than 50% of the time that we spend in the lab is just done analyzing the data that we collected in a relatively short period of time, because that's the biggest challenge.
Do you think AI is going to help us with that, and machine learning? What's your perspective on some of because we hear it in the news all over every day about how fast everything's moving, and it's going to replace this or replace that. What are your thoughts on that analysis and the tools to help us analyze all that data?
Boy, I hope so. Even some of the analyses that we're doing currently, we used to, so for the blood vessels, for example, have to go in by hand and actually physically trace out blood vessels. And we came up with some kind of unique ways to do that using virtual reality systems. But now there's a lot of AI tools that have been integrated into some of the software that we're using already. And they really speed up the process and allow us to trace every single blood vessel and image much more quickly than we could ever have done by hand. And so, that's really been a huge help to us in the lab for sure.
Yeah. Yeah. Your career at Harvard and Mass General started with Dr. Bradley Hyman's lab, and it was one of the major centers there in Boston area looking at Alzheimer's research. What kind of training in that environment? And I know Dr. Hyman, and I think he's an amazing person, and really tries to push innovation and new thought. What, how did that really kind of help you position yourself for your career in your lab and where you're trying to go in the next, couple decades of research?
Well, I think by and large, one of the things that he was able to impart to me was just this ethos of going where the science leads, which I think is really important and not a luxury that everyone has available to them. So typically you end up with funding for a very specific project. And even for me as a postdoctoral researcher, so, initially we had discovered this relationship between tau and blood vessels, but it was just kind of a fortuitous finding that was not the focus of my current project. And so I had kind of set it to the side and I showed him the images and he was like, well, Rachel, those are really interesting. It's like, well, but that's not what we're studying. And so, I was more inclined to set it to the side, whereas he was more inclined to, point out that that was actually really cool. And he was able to support me developing that into more of an idea and taking that into a new direction that otherwise I wouldn't have been able to. And that really has formed the basis of my entire research program now as an independent PI myself. And so, I'm incredibly lucky that he was able to kind of push me in that direction and that we weren't kind of restricted or confined by the, just the atmosphere of funding or, even just kind of the thinking of, you have to accomplish like you have to answer that one question that we set out to answer. And really allowing other folks and who are now members of my lab to kind of pursue their interest and make interesting observations and follow that down the path where it leads them has been something that I've been kind of striving to recreate.
Yeah. Yeah. So tell us about your lab. How big is it? Do you have major focuses that, are sitting up on the wall that everybody's kind of, asking those questions? Give us a visual of what your lab is.
Yeah. So right now it's myself, 2 postdoctoral fellows, and 2 research technicians. And then we have a couple rotating kind of undergraduate students as well. And so the research technicians are post-baccalaureates who have come, and they're usually on their way to a graduate program. So I'm saying goodbye to someone who's moving off to a master's program this summer, and we'll be welcoming a new person to the lab soon. And it's been a really fun environment to be in because we're also in a mix with a number of other PIs such as myself and some clinicians and some academic scholars as well too. So we kind of mix with a much larger group, even though my lab itself is relatively small. And I would say half of the work that we're doing is focusing on experimental systems and using 2-photon in vivo microscopy to just track individual cell changes in the live brain over time and ask questions about how tau pathology relates to things like brain oxygenation and the delivery of red blood cells to the brain, how vascular remodeling takes place, that sort of thing. And then the other half is looking at human postmortem tissue studies. So we're very fortunate to be right adjacent to the Massachusetts ADRC. This is our local brain bank where I have to thank all the donors and their families who contribute this tremendous resource and gift to the research enterprise because we use this every day and it's incredibly valuable to us to be able to not just have access to experimental systems, but also to be able to look at human pathology and really compare what we find in a cell culture system or a mouse model or et cetera to what really happens in a human patient.
Yeah. You had been quoted in an article about the years of failed experiments and, before certain imagery approaches finally worked. Going back to, our previous discussion on that, there seems to be a lot of pressure on a lot of publications, getting things out there. And maybe we're not spending enough time with those failed experiments. what, not to take you down a rabbit hole around politics or today or anything. Can you just, explain why it is really important to fail and not always be successful in everything that you're doing?
Yeah, I mean, every failure teaches us something about maybe a tool that we need to develop that's going to make our life easier in the future or allow us to see something that we wouldn't otherwise have been able to see, or just teach us a lesson about how to perform an experiment better. And so one of the things that we had done was we were trying to track tau neurofibrillary tangles at the cell level in vivo. And it was just a series of experiments where we were trying different approaches, and it was technically extremely challenging and frustrating. And I think in not giving up, it allowed us to observe changes that we wouldn't otherwise have been able to see. And so out of that actually came these observations of blood vessels, which is not what we set out to do, but ultimately like a great discovery, because no one else had spent so much time looking at blood vessels in the brain in relation to tau pathology before. And so we had all these images, and at a certain point you have to just stop and say, okay, here's the proof, and this is what the data is telling us. And I think failure is, for better or worse, just something that you're going to come across time and again in science. But there's always kind of a silver lining to every set of experiments that we do. So
Yeah. And I hope, science is messy, right? Scientists and researchers, they've got to ask these questions and spend the time and not rush to answers. And I think there's a lot of pressure on you sometimes to come to those answers. And, it just takes time. You've talked about this, several times in this discussion about, the cyclic nature of research. You're going in with a hypothesis, you're failing, you need a new, imagery tool or something that wasn't developed, and all of a sudden you've got it. Yeah.
and so often the hypothesis that you set out to test is the complete opposite is what you find too. And then you're just sitting there and you kind of feel like a failure. It's like, oh, I guess I was just horribly wrong. But it's like, well, no, actually, that's really exciting and really cool. And so what assumptions are we making just when we set out to even start thinking about our projects, that we kind of really need to just take a pause and just explore all the different possibilities, right? Because that's where the exciting science really happens.
Yeah, exactly. You're also a Toffler Scholar in 2024, and in 2025, we give out these smaller grants to innovative ideas to early career researchers. How important is that for philanthropy to focus on that cohort of people, especially in today's environment? What did it do for you, or what do you think, that type of support from philanthropy does for researchers?
Yeah, well, earlier you're talking about kind of the pace of the field and technology development. And, for me, what this did was it opened up this possibility to use a technology that otherwise would have been outside of my reach. And it allowed us to do spatial transcriptomics, which is a platform that has kind of recently become available, but it's also very expensive. And so we had some interesting data where we had done a large gene expression study looking at changes in human endothelial cells. And we saw some really interesting changes in AD endothelial cells versus, healthy aging controls. But when you do some of these larger, they were single nucleus RNA-seq studies, you lose all that spatial information. So we don't know, are these endothelial cells next to a plaque? Are they next to a tangle? Where are they in all we know is that they're somewhere in the gray matter, in the cortex, right? So we were able to actually take those findings and then using the spatial transcriptomics platform, which preserves all the gene expression information and a slice of tissue so you can see exactly where each cell is, and really ask some new questions about how those changes relate to disease. And I think it's been just tremendously transformative in our research. It's allowed us to ask questions that we wouldn't otherwise have been able to do. And, it's something that, if we had sat down and written a formal grant and submitted it to the NIH, that would have taken us, a year or more to receive the funding, and then we'd get it started. And, here, maybe 2 years down the road, we'd actually be doing the experiment. So these smaller supplementary grants, they're philanthropically funded, really are tremendously helpful in kind of getting these studies off the ground, because, otherwise it would just be so far delayed. I think that the technology is changing almost every day, and it was extremely helpful.
Yeah. Do you see the philanthropy ecosystem starting to realize that these, if we give to maybe a little smaller money, get it to them quicker, get it to these early career researchers, that we may be able to speed up the process of understanding?
Well, I hope so. I mean, I think I've been lucky to benefit from some of these funding sources. And, I know for me, just, speaking to what I know, is that a lot of the projects probably would have fizzled out really early on otherwise. And I've since been able to take, those seeds, those ideas, and kind of plant them into much larger research projects that have really formed the basis of a lot of the work that my group is doing. And so, I hope it's a trend that continues in the future. I think it's really important for junior researchers, and kind of helps bridge that gap between kind of that postdoctoral stage kind of up through, becoming an independent assistant professor. Yeah.
Yeah. Yeah, it's a tough road. It's a very tough road. You have to learn a lot along the way. That's hard enough, but then now you're having to create your lab and get funding and all of that. It is
Yes.
a definite it's a heavy lift, that's for sure. So, when historians look back into this time and this, kind of this exciting time where we've got AI and new imagery and all that, what do you think they'll say about the scientist? What did we fundamentally misunderstand, or what will they kind of look at and go, oh, this is a turning moment? I mean, we look at this about the genome mapping, right? It's like this opened up everything. What do you think, 10, 15, 20 years from now, people will be saying about this time?
Oh, I think, if you Google just what causes Alzheimer's disease now, I think what's going to pop up is Alzheimer's disease is caused by amyloid beta and tau pathology, and it contributes to loss of otherwise healthy neurons. And I think that definition is going to evolve so much from now to 10, 20 years from now. And people are going to look back and think, oh, wasn't it funny that they thought there was just one type of Alzheimer's disease? I think there's probably many, many different types of Alzheimer's disease, and we're just starting to uncover all those different flavors, and that's really going to guide how we think about treatments in the future.
Yeah. So what is the question I should have asked you that I didn't know I should have asked you, that you want to shed light on, so that people understand what you do and just whatever message that you want to send?
Oh, Deborah, you asked so many good questions, though. I don't think I have any additionals for you, unfortunately.
Okay.
Yep.
Yeah. Well, there's no worry, because sometimes in these things, you take people down a path, and then afterwards they're like, oh, this was really important. I wish I would have said this, or get the message out on this area. So, that's great. We kind of come to our end of our time here, and it's been wonderful, and you're wonderful. How would people get a hold of you if they want to find out more about your research, or if they want to give you money, or if they want to come and join your lab? How do people get in contact with you?
Yeah, you can find me on LinkedIn. That's a great place. We also have a lab website, which is Bennett, Bennett, ALZ lab.com. And feel free to reach out because I love talking to folks about what we do.
Wonderful. Well, thank you so much. This has been fantastic.
Yeah, thank you very much, Deborah. This is great.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.